Okay, let's.
Hi, how's it going? Monday. Hi, how you doing, Amanda? How's it going? Good. It's Monday, my favorite day of the week.
It is?
Why
is it?
Okay.
Actually, I like what I do, so Monday's not bad.
That's good.
How's it going? How's it going? Good. I got all caught up on everything. I told my wife for the first time this weekend, I felt like I've been caught up for the first time in months, and that's a good feeling. I'll get behind today, but you know, as of this morning, I felt good.
Hard to be like, I'm just falling behind.
I'm probably contributing to that too, aren't I? How are you doing? Okay. Yeah, if you want to go back, yeah. So go ahead, you can just plug it in. The main question I have is if I'm getting a hum or not with here. I'll get it. A hum or not with it because my cable varies. There's headphones right there. Okay.
Not here, no hum.
Was that? No hum? Did you hear the tap okay?
Yes, it is that.
No humming.
No hum.
Okay, great. Thanks.
All right.
It's a check because this line is not very good, so sometimes it does. I have to twist it or something. Thanks for checking on it.
Oh, no problem.
Appreciate it.
Have a good day.
Take care. Okay, folks, let's get started. Welcome back. I know you had a fun-filled weekend studying biochemistry. Raise your hand if you did. Oh, this is good. This is good. This is going to be a great term. This is really going to be a great term. Or I'm teaching a bunch of liars. It's possible. I like to think it's going to be a great term. So, last time when I finished the lecture, I left you with a sort of a thought. And the thought was trying to think about how one would determine PIs for amino acids that have more than one, or have an ionizable R group, basically. Okay? So I'll remind you, if we looked at glycine, we saw the following. Okay? So when we looked at the titration curve of glycine, glycine is a very simple amino acid. It only has an alpha amino, and it has an alpha carboxyl group. And they each ionize, one making a negative charge, one making a positive charge. And if we look at the titration curve for that, where we're plotting pH versus hydroxide added, we see something that looks like this. We see two flattened regions, and of course the flattened regions correspond to the buffering regions. And they're centered on the respective pKa values. So the pKa value for the carboxyl group being down here, probably in the vicinity of about 2. pKa for the amino group, the alpha amino group, up here probably around about 9. Okay? And the PI I said was halfway in between those, so that was pretty straightforward. And then I said, well, now imagine that you have an amino acid that has an R group that can also ionize. When you have an R group that can also ionize, what happens? Well, you get 3 flattened regions. And the question is, well, how do you determine the PI? One of the big mistakes students make is they think, well, if I averaged these two to get the PI, then probably what I need to do is just average the 3 of them and get the PI. Okay? And that would be a mistake, because it really shows that you're not understanding what this graph is showing you. So what this graph is showing you is that the PI is found halfway in between the pKa values, around which the charge is 0. Well, how would you know that? How would you know that? Right? Well, it turns out to be fairly straightforward, but most people want to make a harder problem of it than it really is. Let's think about glycine. Glycine has an alpha amino, has an alpha carboxyl. If we look at the charge on glycine, we see it's right here. We put all the protons on. And of course, when we're at the lowest pH, which is way down here on our graph, all the protons are going to be on, because we'll be more than 1 pH unit below all the pKa's. All the protons will be on. What's the charge on that molecule? It's +1, right? The overall charge on that molecule. So I could look at that graph and I could say, well, right down here, the charge on glycine is +1. When I go through a pKa, what's happening? Well, what's happening is I'm losing a proton. After I've exceeded the buffering capacity, I have completely lost the proton. At this point, what's the charge going to be? 0. Okay. And when I go through another buffering region and I pull another proton off, I lose a proton. What's the charge going to be up here? -1. So each time I pull a proton off. Well, I do the same thing with the other amino acids. Okay. So let's think about what's happening with those other amino acids. So if I look at some other examples. Okay, so here is I'll find a more complex one. Yeah, here's some more complicated amino acids. Let's focus on the bottom one at the moment. This is the titration curve for arginine. This one's a little bit more complicated because they don't really show the flattening out. There's actually 3 flattened regions, but they just kind of drew it as a big diagonal, which is kind of dumb for teaching purposes. So we'll focus on the bottom one. Here we can clearly see 3 flattened out regions, each one of them corresponding to a pKa. Arginine is an amino acid that has an amine group in its R group. If I put all the protons on this guy, what's his charge going to be? Add them up. +2, right? A plus on the alpha amino, a plus on the R amino, and a 0 on the carboxyl group. So when I start down here, the the charge in the molecule is going to be +2. What's the charge going to be right here? 1. What's the charge going to be here? 0. And you notice, look where the PI is, right where the charge is 0. Now, another way to look at this is if we look at the charges, this is sometimes a little hard for students to envision. If we look at the charges at the pKa values, we can consider those. What are the charges at the pKa values? Well, let's go right here. What's the charge? If the charge here is 0, what do you suppose the charge is at that pKa value? Is it 1? It's not 1. Why is it not 1? That's right. Half these guys have the proton on, half have it without. Your mistake in thinking about these is going to be you're thinking about these as individual molecules. How can a molecule have a charge of plus 1/2? It can't. But half the molecules can have a charge of 1, and half the molecules can have a charge of 0, so that the overall net charge is the average plus 1/2. So here, the charge is essentially plus 1/2. What's the charge up here? Minus 1/2. Okay? So we can approximate the charge by approximating the average charge of a molecule in the solution. The average charge of the molecule in the solution here is plus 1/2. The average charge of the molecule here is minus 1/2. What do you suppose the average charge of the molecule is right here? Plus 1.5, right? We put another proton on it. Okay. So you get the idea for these. So if that's helpful to you, fine. If not, then you can just go through and sort of count the charges and figure out how these things go. The mistake you'll make is if you don't go and figure out where the charge would be 0. You try to average it, or you try to do some, say, oh, well, the charge up here was 2 up last time, so it's probably 2 up this time, so therefore I know what the charge in this molecule is. You'll end up in trouble if you do that. Okay, so don't do that. Okay. Questions on that? You guys are really dazed today. What is this? Was the singing that bad on Friday? That's where you get rumble. You mentioned the word sing, all of a sudden it goes, you hear it going all across the room. All right. Let's see. So let's get off that. Let's now actually talk about the individual structures that we see in proteins. So we now know what I've just been talking about are the composition of the amino acids that go into proteins. Okay? The composition of amino acids that go into proteins, there are 20 of them, as I said. Those amino acids go together, and they are joined together to form peptide bonds. So the way that amino acids are joined in proteins are through peptide bonds. The peptide bonds are formed in ribosomes during the process of translation, which we'll talk about next term. So translation joins the amino acids together, forms peptide bonds. And you can see the peptide bond being formed here. A peptide bond is formed when an alpha carboxyl combines with an alpha amino to make this bond right here. That results in the splitting out of water. And the splitting out of water, okay, is an essential part of that process, resulting in a covalent bond between the carbonyl group and the amine group. Okay? Now, notice when we split out water, we did something interesting. What we did was we eliminated an ionic charge, and we eliminated an ionic charge. Thus, the peptide bond, when we go to calculate, if we were to try to calculate the overall charge of this molecule, we would only have to consider the charge of the alpha amino, which is still free, the alpha carboxyl, which is still free, and any of the R groups, which are out here. We would not have to consider anything relative to the peptide bond. Well, for one peptide bond, that's okay. But if you have a molecule that has 50, you just save having to calculate 50 charges. That's pretty good. That's pretty cool. So wherever you have peptide bonds, you no longer have to worry about calculating charges of those guys because they don't, the peptide bond itself is not charged. Everybody with me? That will become important, and the TAs will talk about that not this week, but actually next week, when they, and I'll talk about this a bit as well, when we talk about calculating the charge not of amino acids, but now of polypeptide chains. Calculating the charge of polypeptide chains is actually simpler than it would seem because of what I've just shown you right here. I've also got a video on the webpage of me talking about that I will encourage you to take a look at. Okay? And you can listen to that. If you have questions about how to calculate charges of polypeptides, you should also let me know. Okay, now, peptide bonds are essential. Peptide bonds hold the amino acids together. And peptide, excuse me, peptide bonds are arranged in interesting ways. This shows a polypeptide chain. And unfortunately, your book was stupid enough to pick two different glycines in the middle, so you don't really get a good sense of all the different possible R groups that could be out there, but that's okay for the moment. What I want to show you is how, when we look at that polypeptide chain that's just been formed, I want to look at how those R groups arrange themselves, how they're arranged around that protein. Look at this. Here's an R group over here. Here's an R group down here. Here's an R group up here. Here's an R group down here. Here's an R group up here. Do you see the pattern? The R groups are generally arranged so that they're on opposite sides of the polypeptide chain. Why do you suppose that might be useful? Steric hindrance, you bet, because some of these R groups are going to be big, and keeping them away from each other is a consideration. Now this is a simple representation, okay? And in reality, these guys are in three-dimensional space, not in two-dimensional space, so there are other considerations. But suffice it to say that the arrangement of these R groups, as they're shown here, is very, very useful. These R groups are in fact arranged in what's called cis-trans configurations, thanks to the cis-trans arrangement of the alpha carbons to which they're attached. And I'll show you that in a second. Let me repeat that. Okay? So the R groups are arranged in a cis-trans configuration because the alpha carbons that they're attached to are arranged in a cis-trans configuration. I'm going to show you another figure for that that's a little bit better, I think, in just a second. Another thing this figure shows you that I want to point out is when we look at polypeptide chains, and by the way, I interchange the terms polypeptide and protein. Technically, there are different things, but in this class, I refer to them as the same thing. So when I'm talking about a polypeptide, I'm essentially talking about a protein. Okay? And as I said, technically they are different things, but I'll use the terms interchangeably. When we talk about a polypeptide chain, we can always talk about different ends of it. A polypeptide chain will always have a free alpha amino, there's the very first one, and a free alpha carboxyl, there's the very last one. And the reason I say first to last is because polypeptide chains are made starting at this end and going this way. They're made in what's called the amino carboxyl direction. So we have an amino end and we have a carboxyl end corresponding to the location of the alpha amino group and the alpha carboxyl group. Okay, now let's look at that arrangement of these guys in cis-trans configuration. And this figure shows a little bit better how those R groups are oriented. Here, for orientation, is our peptide bond. That's the carbonyl group and the nitrogen. The alpha carboxyl and the alpha amino group. We're right there. There's the alpha carbon. There's the alpha carbon. Notice how the alpha carbon is up, how the alpha carbon is down. The alpha carbon is up, the alpha carbon is down. The alpha carbon is up and down. Okay? Now there's a reason we draw it this way, and I'll show you in a minute, that will, I think, also hopefully help to orient you with respect to why we think of these as cis and trans. There's a very important reason. You remember when we have cis and trans and you had organic chemistry, what did you have to have cis and trans? Chemists, where are you? What do you have to have cis and trans? No, you have to have double bonds, right? Cis and trans are orientations around double bonds. And in fact, with the double bonds, do you see any double bonds in here? Well, you see a double bonded carbon to oxygen, but that's not a double bond. There's no double bonds showing between the carbon and nitrogen backbone. Another thing I want to point out for you is that when we look at a polypeptide chain, notice we have alpha amino, alpha carbon, alpha carboxyl, alpha amino, alpha carbon, alpha carboxyl. Okay, the polypeptide backbone is very simple. Amino, alpha carbon, alpha carboxyl. Amino, alpha carbon, alpha carboxyl. That's the backbone of the polypeptide chain. What makes the polypeptide have its characteristics are the chemical composition of the R groups that are sticking out. And that's why knowing R groups is important. I've had several questions. What do we have to know about structure? Okay. What you have to know about structure is the categories they fit into. You need to know, does, for example, Does this have a carboxyl group? Does this have an amino group? Does this have a sulfhydryl group? Those are all groups that can ionize. So you need to know those. You don't have to draw them, but you need to know what they are. You need to know what those charges are when they ionize. Carboxyl being -1, amino being +1, sulfhydryl being -1. Okay? You need to know those, but you don't have to draw them. You don't have to draw the structures. Okay, now, I promised you where we would get cis-trans. Where do we get cis-trans? We get cis-trans from resonance. Resonance basically involves the shuffling of electrons around bonds, as you can see here. And peptide bonds have what's called double bond character, because just as we can draw the electrons around this in a carbonyl group as we show here, so too can we swap those electrons around and put a double bond in there that looks like this. And it turns out that peptide bonds have resonance, meaning that they behave most of the time like they are double bonds. They behave like they are double bonds. Now, when we look at this, what do we see? Well, now the cis-trans becomes very apparent, because here, this guy is shown in a trans configuration, this guy going up, this guy going out that way. So the orientation of the alpha carbons around that peptide bond are in a trans configuration. The vast majority of the time, and by vast majority, probably on the order of several hundred to several thousand to 1, the vast majority of the time, the peptide bond is oriented with the alpha carbons in the trans configuration. Now the next figure I'm going to show you will hopefully show you that, and that corresponds to, not that one. Here. Here is a trans configuration where we orient the alpha carbon down and up around that peptide bond. Okay. Here is what it looks like if we put it in the cis configuration. Now that steric hindrance that we talked about before being a consideration becomes very apparent, because now if we have this in the cis configuration, what we see is that in fact we have a lot of possibilities for our groups to get into each other's face, and because of van der Waals forces, we cannot put them too close together, and that's a consideration. So we can't put them too close together. So it's for this reason that peptide bonds orient themselves, for the most part, in the trans configuration. The cis configuration is not very stable. Okay, we won't worry about bond lengths. Prolene is one interesting sort of exception. Prolene still strongly prefers to have its bonds in the trans configuration, but proline is one of the amino acids that doesn't have it as abundantly in the trans configuration as do the other amino acids. So whereas the other amino acids may have several hundred or several thousand to 1 preference for the trans configuration, proline may be even slightly under 100 to 1, but it's still strongly preferred, just not as strongly preferred as it is for the other amino acids. Proline, I will remind you, is the amino acid that has that ring, and I said that there are some unusual things that happen as a result of proline having a ring, the ring including the alpha amino group, as we've seen before. Okay. Now, well, I've been talking so far about bonds that don't rotate. A peptide bond, if it's double bond, really doesn't rotate. A double bond is pretty much a fixed thing, which is why we have cis and trans. Okay? But a peptide bond is only one of 3 bonds that actually occurs in the backbone of a polypeptide chain. It's only one of 3 bonds in the backbone of a polypeptide chain. The other 2 bonds have names. Okay? They're called phi, as you see on the left, and psi. Right? Phi is the bond between the alpha amino and the alpha carbon. Psi is the bond between the alpha carbon and the alpha carboxyl. Now, what are phi and psi? Well, phi and psi turn out to be interesting things. I need a couple of pieces of paper. Can somebody give me a couple pieces of paper that you got loose? Don't tear something out. Do you have loose paper? Anybody? Okay, here we go. 2 pieces of paper. All right. Now, when I think about a peptide bond is flat. A peptide bond defines a plane. A piece of paper is a plane. Okay? It's a flat thing. Right? So if we look at this peptide bond that we have between, let's say, the nitrogen in blue and the carbonyl group, that is, this is one peptide bond right here, and here's another peptide bond over here, the two bonds in between there are the phi and psi bonds. Those guys are single bonds. They're not double bonds. And those guys, if you remember from your organic chemistry, those guys can rotate. Well, what does that mean? Okay? It means that whenever I look at an alpha carbon, the alpha carbon is the thing that's in the middle, the alpha carbon is between two peptide bonds. The alpha carbon is between two peptide bonds. The alpha carbon, of course, the one that has the green R coming off of it. Here's one plane corresponding to one peptide bond. Here's the other plane corresponding to the other peptide bond. And my nose is the alpha carbon. Okay? Now, this is not spatially accurate, so you'll have to use your imagination a little bit. I've got a plane out here. I've got a plane on either side of my nose. And those planes, they rotate as a flat surface. The whole surface rotates because, remember, we can't rotate within the plane because we've got a double bond. It rotates as a flat surface. Everybody with me? Phi on one side, psi on the other side. And what phi and psi correspond to are the rotational angles relative to 0. We won't worry exactly about where 0 is. That's not important. But it is important to recognize that they are rotational angles. We could imagine, for example, that if my nose is that alpha carbon, okay, and I have these two planes right here, there are going to be certain rotational angles that are going to work and certain rotational angles where there's going to be steric hindrance. You with me? So certain rotational angles will be favored, other rotational angles will be impossible to exist because they crash into something. In this case, my head. In reality, what they do is they crash into each other. Okay? Now, what we've just defined with phi and psi are a way to describe the structure of a protein. A protein can be thought of as a series of alpha carbons, each of which have a phi and psi angle associated with it. So we can define the structure of a protein by defining all of the phi and psi angles for all of the alpha carbons. Okay? Whoop-de-doo, right? If you're into geometry, if you're into modeling, if you're into placing atoms in space, this is a very, very powerful tool. Because I've now positioned every plane of the polypeptide chain relative to an alpha carbon all the way as it goes dancing through the molecule. Every one of them, I know what it is. That's very cool. Okay, thank you for that. Now, as I told you that we can imagine that there are many angles that may not work because things are going to come crashing into each other because some of those R groups aren't going to fit, et cetera, et cetera. Big things aren't going to fit. Well, there's a famous Indian scientist named Ramachandran who recognized this long ago and said, you know, it's not rocket science. It's protein science is what it is. He says, it's not rocket science. We could take a computer and we could look at all these bond angles because we know the distances between everything, and we could actually predict which angles are going to work and which angles aren't going to work by predicting where things start crashing into each other. So he made what he described, what we now call a Ramachandran plot. And a Ramachandran plot looks like this guy here. Ramachandran plot plots phi on the x-axis against psi on the y-axis. Both plots include a full 360 degrees, minus 180 to plus 180, and minus 180 to plus 180. And again, our orientation of these really isn't critical for our purposes. If we're doing biophysics, orienting everything would make a lot of sense. I'm trying to illustrate general principles here to you. Okay? What he did was he said, okay, computer, here are the side chains, here are the sides, here are the bond angles, And I want you to start rotating this thing through space, and we know when things get too close that they're not going to fit, they're going to repel each other, so these won't work. Tell me all of the regions of rotation that are stable. And that's what the Ramachandran plot shows you. The stablest regions on his plot are shown in green. Darker the green, the more stable. The lighter the green, the less stable. And the more white, wherever there's white, it's unstable. Now, no, I'm not going to ask you on there, is 120 degrees by minus 60 degrees stable? That's not the important message here. The important message, there's a couple of them. The important message is, first, that there's a lot of rotational angles that are not stable. There are limitations where those rotations can occur. The second, as we're going to see, is that these stable regions that we see on a Ramachandran plot actually correspond to real-world protein structures that we know a lot about. It turns out that the Ramachandran plot, which was theoretical, turns out to be a very, very useful tool for predicting protein structure. And it allows a scientist, when they're trying to build a model of a protein, you guys have seen in books, you've seen three-dimensional models of proteins. How do those three-dimensional models come about? Well, one of the ways they come about is by people looking at data and saying, okay, this looks like this could be a rotational angle of so-and-so, so-and-so, etc. Putting those phi and psi angles together. And if you know what the limits on those phi and psi angles are, you have a great head start because you know what not to use. So Ramachandran plots are very, very useful for that purpose. Now I'm going to show you some aspects of the Ramachandran plot in just a minute. But before I do that, I want to stop and take any questions or concerns that you have about that. Questions? You guys are really a silent group. Okay, there you go. Jessica. Their structures can be described in that way. Yeah. Yeah. But it's a little hard to get your head around because you start thinking about, well, there are hundreds of these in proteins. In some cases, thousands of these in proteins. Okay? But the reality is that you're describing the overall structure. When a scientist goes and they do X-ray crystallography, okay? That is, they're shooting X-rays through here to help determine what the structure is. They're going to determine the orientation in three-dimensional space, that is the position in three-dimensional space, of over 100,000 atoms. Okay? That's not a trivial thing to do. Any help that you have along the way in helping you to position those and knowing what the rules are, Ramachandran plots give you a real good notion of what those rules are in going through and doing that. Yes? Yeah, it's quite complicated. I will actually talk about X-ray crystallography probably either later this week or early next week. And fortunately, I'm not going to make you interpret X-ray crystallography. It's a very involved and complicated process involving a lot of computational work. But basically what you're doing is you are interpreting where X-rays come out of a crystal after you shoot X-rays into them. And I'll say more about that at that time. Yes? Uh-huh. So by the time you're doing that, you already know what those R groups are. So his question was, can you not only determine where they are, but what they are? And the point is that by the time you've gotten to that point, you already know the complete sequence of amino acids of that protein. Yeah. Okay, now that brings me actually as a good sort of transition to the next thing I want to talk about, and that's sequence. You're going to hear a lot about sequence. What is sequence? The sequence refers to, in the case of a protein, the order of amino acids. The order of amino acids. So if the first amino acid in a protein is methionine, and by the way, methionine is almost always the first amino acid in a protein. If the first amino acid is methionine, and the second amino acid is aspartic acid, and the third amino acid is leucine, that's going to have a different structure than a protein that starts with methionine, and then has tryptophan, and then has valine, for example. Why? Because those R groups are different, the structures are different, The way they orient themselves are going to be different. The chemistry is different. Everything will be different. So what I'm trying to tell you is that the sequence of amino acids, we'll come back to this again and again, but the sequence of amino acids of a protein gives the protein all of its properties. We're going to talk about secondary structure, we're going to talk about tertiary structure, we're going to talk about quaternary structure, we're going to talk about catalysis, we're going to talk about Vmax, and we're going to talk about all kinds of things with respect to proteins and enzymes. But everything, every single thing about a protein comes down to its sequence of amino acids. If we change that sequence of amino acids, we will change the structure of that protein, change its ability to do things, etc. Mutation changes amino acid sequences. That's what happens. Yes, question? I'll talk about chaperone proteins a little bit later when we talk about folding. What's your question? Chaperone proteins do not have a greater effect than sequence. Sequence is the thing and is the only thing. All chaperone proteins help a protein to do is fold, but that folding is dependent upon the primary sequence, which is what we're talking about. Okay. Now, so I need to define for you, therefore, a very important term. The sequence of amino acid is what we call the primary structure of an amino acid. The sequence of a protein is what we call the primary structure of the protein. So all this time I've been talking about primary structure. I've been talking about the sequence of amino acids comprising it. Now the sequence of amino acids is specified by the genetic code, and we'll talk about that when we talk about that next term. Bottom line, sequence is not something that we take lightly. All right, so primary structure of a protein is its sequence. We have to look at another level of structure of a protein. We're going to talk about 4 levels of structures of proteins. The second level of structure of a protein is what we refer to as the secondary structure. Well, if the primary structure is the sequence of amino acids, what is the secondary structure? Well, I've sort of alluded to it already. I've said that you can imagine that we've got R groups that are out there that are having to position themselves so they don't interact more than they need to. Maybe in some cases they might even like to interact. Maybe they've got a plus and a minus next to each other, something like that. So we can imagine that there will be some local effects of the R groups on each other. So when we talk about secondary structure, we start thinking about local effects. These local effects are what I define as interactions between amino acids that are close in primary sequence. What does close mean? Let's say less than 10 apart. Secondary structure arises from interactions between amino acids that are close in primary sequence. Now, when we look at this, we see primarily 2 main structures. 2 main structures. The first of these are known as the alpha helices. And by the way, the structures I'm getting ready to show to you were discovered by Linus Pauling back in the 1940s. And it was these structures for which Linus Pauling won his first Nobel Prize. Linus Pauling being the most famous scientist ever to graduate from Oregon State University, and being the person whose name is on the new Linus Pauling Science Center that's being built on the west end of campus. Okay. The alpha helix was discovered by Linus Pauling. The alpha helix is a regular, repeating structure. It's a helix. Now, it's not like a DNA double helix, because a DNA double helix has 2 chains wound with each other. An alpha helix has a single chain, a single polypeptide chain. The structure of the alpha helix was known, in fact, before the structure of the double helix was known. Okay? The alpha helix arises from interactions that are happening among amino acids that are relatively close to each other in primary sequence. I'll show you what some of those are in a second. These are different representations of the alpha helix. That's a little hard to see, isn't it? But what this shows you is you see some of those interactions. You see those dotted lines right there? What do you suppose those dotted lines refer to? Hydrogen bonds. You got it. Carbonyls are interacting with hydrogens on amine groups. If we look down an alpha helix, that's what we see. Anybody see anything interesting about that structure besides the fact it's got a hole in the middle? All the R groups are pointed outwards. Okay? The R groups are the big guys. The R groups are orienting themselves so that they're pointing away and outwards. Now, in spite of that, some R groups are going to be too big to fit very well with an alpha helix. When that happens, they get a bulge, and you disrupt the helix. If we imagine phenylalanine or tryptophan, those have pretty good sized R groups. In some cases, those are going to disrupt an alpha helix. Another thing that's going to disrupt an alpha helix is proline, because proline doesn't have flexibility. To make a helix, you've got to rotate bonds, and you've got to get things oriented in the proper way. Because proline can't rotate, proline doesn't form alpha helices very well. Okay, we'll see a little bit more about that in a bit. Okay, there's a sort of space-filling representation. All right, now hydrogen bonds are the primary forces that help to stabilize alpha helices. You can see the interactions as shown in this two-dimensional plot where the red arrows show the interaction. The interaction is possible because, again, it's coiling around, and so something that's a few amino acids away, when the coil comes around, has that opportunity to interact again. That's what we're seeing in these guys here. Okay? When we look at the Ramachandran plot and we discover where the alpha helices fit, looky there. In two of the theoretical regions, we see that, in fact, the right-handed helix, which is by far the most common, occurs, and the left-handed helix, which is quite rare, is also in that theoretically predicted area that Ramachandran saw. Okay? It tells us that theory actually tells us something about the real world in some cases. Yes? They are one for each amino acid. So we're actually doing this as if it's a polymer of amino acids. You're exactly correct.
So is this a superposition amino acid plots?
So what this is, and when you look at the red region, what they've done is they've gone to the X-ray database where they know the actual orientation of every single one, and they are superimposing that on here. But you're exactly right. Yeah. So they've said here's a phi and psi angle, irrespective of what these R groups are, and this is in an alpha helix, and when you do that, you see everything fits in here. But you're right. Very good point. Okay? All right. So that's the alpha helix. The alpha helix is a very common structure that we see inside of proteins. When we look at a protein like ferritin, we discover ferritin has a three-dimensional structure that looks a little bit like this. And ferritin is a protein we'll talk about later in the term. It's a protein that binds iron. And look at this. Alpha helix, alpha helix, alpha helix, alpha helix. What do you suppose that little coil is right there? Or that little coil right there? It's not alpha helix, is it? Okay. What it is something that doesn't form a regular structure. When we talk about secondary structure, we're talking about regular repeating structures. An alpha helix is a regular repeating structure. This is something we refer to as what's called a random coil. A random coil is going to have in it amino acids that are not consistent with an alpha helical structure. They're going to go do their own thing because they won't obey and they won't fit in the confines of an alpha helix. It turns out that they're very, very, very important, however, in understanding protein structure. Because the overall structure of this guy, of this protein, is a function not only of the alpha helix, but how the alpha helices are arranged relative to each other. That arrangement is due to the random coil. Yes? Okay, so the question is, are there only certain sequences that will form an alpha helix, or are there multiple possibilities? The answer is that there are many, many sequences that will, and there are numerous examples where it won't. So the numerous examples where it won't tend to be clusters of amino acids, and particularly amino acids, like I said, that either have bulky R groups or like proline that cause folds or other considerations. It's a complicated answer to your question, but the reality is that there's a lot of things that will form alpha helices. Okay? Yes, sir. No. The alpha helix shape is due to the composition of the amino acids facing it, and it's basically the same repeating structure over and over and over. The arrangement of the alpha helices with respect to each other. So this guy is positioned right where it is because this random coil here is the length that it is, and it's in the position that it is. If I shorten that random coil, then this isn't going to have the same structure. It's going to be more bunched together. So the random coil simply orients the individual helices with respect to each other. Yes, Lori. The random coil is technically not secondary structure. Only repeating structures are secondary structures. I'm going to show you one other repeating structure in a second. Good questions. Okay. Now, so that's your first view now of the three-dimensional nature of a protein. That's actually a fairly simple one. We'll see some much more complicated ones later. Another repeating structure that we see in proteins, besides the alpha helix, is something known as the beta strands. Okay? Beta strands, interestingly, not surprisingly, appear in the other stable portion of the Ramachandran plot. Beta strands are what's called a two-dimensional helix, because basically they look like folds of a curtain. All right? They look like pleats, up, down, up, down, up, down, up. Kind of like we drew that backbone. That's what pleated sheet or beta strand. Let me back up. They're called beta pleated strands look like. You're seeing, as I'm starting to call them sheets, that I slip into the same problem that your book and others fall into, and that is if you put together multiple beta strands, you create what's called a beta sheet. And so people mistakenly will refer to beta strands as beta sheets sometimes, when technically a beta sheet is only a compilation of several beta strands together. Okay. So beta strands, I'm going to show you a structure in a second, but beta strands have this portion of the Ramachandran plot. And when we look at them, they look kind of like that pleated sheet, and not that different from that structure that we originally drew for the cis and trans nature of the R. There's the R group down, R group up, R group down, R group up, etc., looking like that. The difference between this and the helix is a three-dimensional structure. The strand is basically a two-dimensional structure. It doesn't fold out, it doesn't coil out, but it just goes up, down, up, down, up, down, up. So it's a two-dimensional representation of a helix. Here are some illustrations, and don't worry about what it means by parallel and anti-parallel. That's not the most important thing. The most important thing is you can see a beta strand on the top and a beta strand on the bottom, and you see interactions between them, and what do you suppose is the nature of those interactions? Hydrogen bonds. So now you begin to see how you can orient these guys in the formation of sheets. Okay? If we look at something like silk, silk is composed of a lot of beta strands to make sheets. That's what silk is made of. Yes, Jessica. Yes, you need to have two or more to make a sheet. Yeah. Okay. Okay. Now, let's see. Where am I at here? There's another representation. These are what are called parallel versus anti-parallel. It's not critical for our purposes. But again, you see hydrogen bonds are stabilizing these guys. And now here we have parallel on the top and anti-parallel on the bottom and blah, blah, blah. But the important thing is that they're interacting with each other, and we have this folding nature that's there. Interestingly, beta strands can come together in interesting ways. That's not a very good representation, but there's a better one here. Okay. And you can see that in this case, the blue corresponds to the beta strands, and they're interacting with each other, and they're actually forming what's called a barrel. All right? So they've actually formed a circular structure due to their interactions that sort of you see twisted like this. This protein you see has both beta strands, it has one alpha helix, and it's got a bunch of random coils. You should be able to draw this on the exam. Okay, you won't have to draw it on the exam. All right. Turns. All right. Well, what makes up random coils? All right. Random coils happen because some amino acid in the sequence is just really not very compatible with an alpha helix. You say, well, that should be known. Which ones are known? Well, it kind of depends on their interaction with the neighboring ones. And it's a complicated picture. It's not just necessarily the next one, but it could be the next 2 or 3 or 3 or 4. So it's not easy to say it's always this amino acid. But we can look at some patterns, and we can see some patterns. One of the patterns that we see is that proline doesn't fit very well in alpha helices. Proline frequently forms what we call a bend. Some of those random coils that you saw there were actually bends that were occurring that were disrupting the alpha helix. The bends start with a proline, and then these are the next amino acid, next amino acid, next amino acid. So these actually favor, okay, the disruption of the alpha helix, the disruption of the beta strands, and making something that now is a random coil. Proline is very commonly involved in that. Another amino acid is very commonly found in these bends as well, and it's glycine. Anybody have any idea why glycine might be important in these bends? What's that? Less steric hindrance. You got a lot of steric hindrance with proline, and then you got a lot of flexibility with glycine, so you put the two together and you can actually do something that you couldn't otherwise do. Proline and glycine come together nicely to make bends in many cases. Okay. Loops. There's a loop. That's basically what we saw before, the barrel sort of structure. Now, what I want to finish with today is talk a little bit about fibrous proteins. So fibrous proteins, here are some representations of fibrous proteins. Real good example of fibrous proteins, your hair. Okay? Your hair is made of a protein called keratin. And keratin is comprised of a sequence of amino acids that almost exclusively makes alpha helix over and over and over and over and over. It doesn't have bends. How do I get curly hair, right? Isn't that what curly hair is a bend? Well, it is in a sort, but not the sort that we're talking about here. These bends that we have, that we talk about here, are flexible, right? So they're not going to just stay in one position and be curly. So curly hair doesn't arise from that. Curly hair arises from the fact that part of the sequence of the alpha helix that you have contains cysteine residues. Cysteine is the amino acid that you recall has a sulfhydryl group on the end. Sulfhydryl, of course, is an SH, and it's very reactive. I mentioned that before. The cysteine up here can interact with the cysteine down here and form a covalent bond, and now you've got a real band, and that real band is held by a covalent bond that's going to be hard to break. That's where curly hair arises from. When people go and they get a permanent, and they come away and they stink, the reason they stink is in the hair shop, they had used a chemical that had broken those disulfide bonds and rearranged them in a way that the hairdresser wanted. Okay? It's not permanent in the sense of being permanent, but it'll last for a while. And to break disulfide bonds, you have to use sulfury type chemicals to do that, and those sulfury type chemicals stink. That's what happens with a permanent. People don't get permanents nearly as much as they used to. Okay. A fibrous protein is a protein that has only secondary and primary structure. Only secondary and primary structure. Okay? I mentioned earlier that silk is comprised of protein that has basically beta sheets over and over and over and over. Okay? So some, like keratin, have only alpha helices. Others, like silk, only have beta strands. Here is a very interesting protein. I'm going to finish. I've got about a minute or so, and I'll start with this next time as well. I'm going to finish with this. This is the most abundant protein in your body. The most abundant protein in your body is collagen. Collagen is literally the glue that sticks you together. It holds cells together. It holds tissues together. It gives you the ability to stand up. Without collagen, you're in deep doo-doo. Collagen is an unusual helix in a couple of respects. It's a triple helix, and it's an unusual set of helices even there. If we look at the primary structure of collagen, we discover something quite surprising. Anybody see any patterns in there? glycine, proline, glycine, proline, glycine, proline, glycine. That stands for hydroxyproline, glycine, proline, hydroxyproline, hydroxyproline, proline. Look at this. I've been sitting in here telling you that proline isn't very consistent with making helices, and now I'm trying to tell you that it makes helices. You guys aren't going to believe me again, right? Okay. It turns out that there's two things that contribute to this. One is the fact that glycine is very abundant. You see glycine in all the reds there. The other is that the modification of proline also facilitates this. And I'm going to talk about that next time. The hydroxylation of proline is absolutely critical for the function of collagen. Talk to you about that on Wednesday. See you guys then. Hey, guy, let's go. Hey, Panda. How you doing, Becky? Good. How's your classes starting out?
Pretty good.
Good.
That was really interesting.
Oh, is that right? Yeah. Come sit in on my lectures. You're welcome.
Okay.
Yes.